In today's video, we're going to be looking at oligosaccharides and polysaccharides. This is a continuation of my last video on carbohydrates that looked at monosaccharides and disaccharides, and is a continuation of the AQA A-level biology topic on carbohydrates and biological molecules. I would really strongly suggest that if you haven't seen the first video already, to go back and watch it before we continue on to this one, as it covers some very important stuff about bonding and condensation reactions. So we'll start with oligosaccharides. Now, we don't need to know a lot of these for A-level biology, as they're not really covered too much in the AQA course, but it is important to know that they exist and a few little bits about what they do. So oligosaccharides are comprised of around 3 to 10 monosaccharide subunits, so 3 to 10 small sugars, and they have roles in cell recognition and cell binding. So we've got an example here on this top right hand corner, and this is raffinose, and this is a trisaccharide, so has 3 monosaccharide subunits, and is comprised of galactose, glucose and fructose, which are the 3 monosaccharides that we need to be aware of for this course. That's pretty much all I'm going to cover on oligosaccharides. We don't really need to know much about them, so we're going to move on to polysaccharides now. It's a much more complex and in-depth topic that we need to know about. So polysaccharides have multiple roles. Here we can see an example here that's just popped up on the screen. So one important role that they provide is structural support, and we'll look more at this when we cover cellulose later in this video. And they're also an important energy storage molecule, and we'll see this when we look at glycogen and starch. And they can form multiple different structures, so they can form these coiled structures, which are very compact, very easy to store, but they can also form these amazing branch chain structures with thousands of branches coming off from a single chain, and this is very important for energy breakdown. So we'll start by looking at glycogen. Glycogen is the energy storage material for animals. You can see a picture here in this top right hand corner. You can see it's got all of these side chains. It's a big molecule, and this is very important when we look at how it's broken down. So glycogen is a polysaccharide formed of excess alpha glucose. So if you ate a really sugary meal and had lots of excess glucose, then it would be stored as glycogen. A process called glycogenesis would occur, which is where glycogen is formed and it's then stored in the liver. Now, the presence of these side chains allow for a really fast release of this stored glucose. So this is a process called glycogenolysis, which literally means splitting apart of glycogen. And it is broken down into these glucose 1 phosphate units, which are then used in producing energy, and we'll cover this in another video. It's a second year topic, so it's not something that is necessary to learn at the moment. But these side chains, they allow multiple points for enzymes to act on, so it can cause a really fast release of energy. It's a very compact molecule, so it's very easily stored. Like I said, we can have large amounts of it stored in the liver, and this is partly why athletes do something called carb loading. You see it with marathon runners. The night before they run, they'll eat a lot of carbohydrate-based products, and they'll get this good storage of glycogen in their liver, so that when they're running and they've used up their initial energy reserves, that this glycogen can then be broken down to provide more energy. And so if you don't eat breakfast one morning, your body will break down the glycogen that's stored in your liver, but once this is all gone, you'll start to feel very hungry, and then your body has to look for other energy sources to use. A glycogen is also an insoluble molecule, and this basically means it can't really dissolve in water, and this is good because it has no effect on water potential, and this would be a massive problem if it did impact water potential, because when glycogen is stored in the liver, this would cause water to enter by osmosis, and then your liver will become very inflamed, and you suffer from something called hepatitis, which, if we break it down, hepato is the liver, and itis means to become inflamed. So if it did have any impact on water potential, it could cause some quite serious side effects. So we're going to move on to starch now. Starch, again, is a polysaccharide, but it's formed by plants. It's the energy storage molecule in plants, and again, it's formed from excess alpha glucose. It's a compact molecule, so it is easily stored, similar to glycogen, and again, it's insoluble, so it has no effect on water potential. And again, this is very good for plants, because if it made starch and then caused a load of water to enter the cell would swell and it would burst, and it'd have cell lysis, and that would cause a whole load of other problems. So starch typically forms one of two structures. So the first one we'll look at is amylose, which we can see here in the top right hand corner of the screen. And amylose is an unbranched structure, and it forms this coiled structure. And it's a very compact molecule. These coils allow it to form these very small compact structures, so it can be very, very easily stored. And the reason why it coils is it forms something called a 1-to-4 glycosidic bond. So that is a glycosidic bond that forms between carbon 1 in one monosaccharide subunit and carbon 4 in another unit. And amylopectin is the other structure that starch essentially forms. And amylopectin is a branch structure. Now, similar to glycogen, this allows it to be rapidly broken down. So we can see amylopectin in the bottom right-hand corner now, and you can see it looks very, very different to amylose. It has all these branches coming out of it. And again, this allows enzymes to act on it rapidly and break it down to release glucose. Now, it forms this branch structure because this time it forms 1-to-6 glycosidic bonds. So there's a glycosidic bond formed between carbon 1 in one monosaccharide unit and carbon 6 in the other. And this gives it very different properties. So amylopectin is obviously probably the one that would typically be broken down first if your plant was in desperate need for glucose. But once these reserves run out, then amylose would then start to be broken down. So the formation of polysaccharides is very similar to the formation of disaccharides that we looked at in our last video. So here we can see we have a long chain of alpha glucose units, and 4 of them have joined together to form a polysaccharide. And we have a 5th single alpha glucose subunit here. Now, similar to what we looked at last time, but just to recap, this alpha glucose will bond to the end of the chain, and we can see that a glycosidic bond has formed. Now this structure contains multiple glycosidic bonds, so we can see that there's 4 of them. Makes sense, we've got 5 alpha glucose subunits and they've all bonded. So we have 4 glycosidic bonds. We also need to label that we have 4 water molecules. So each time a new subunit gets added into this structure, it will form 1 new glycosidic bond and release 1 new water molecule. Now, a good way to remember how much water has been released is to look at the number of subunits excuse my handwriting, minus 1. So there's always 1 less water molecule released than the number of subunits that are present within the polysaccharide. So we can see here the subunits, we've got 1, 2, 3, 4, and 5, and our number of water molecules, we've got 1, 2, 3, and 4. And this makes sense of our equation. Number of units, -1. We've got 5 units, -1. That makes 4. And there are 4 water molecules given off when this polysaccharide is created. Now, this is an important thing to note and to remember, because you may get an exam question that says something along the lines of a polysaccharide is formed of 12 monosaccharide subunits. How many water molecules are released? And you can say 12 subunits, -1, 11 molecules of water. And the same thing if they were asking a question on hydrolysis. A polysaccharide consists of 17 subunits. How many molecules of water would be needed to split it completely into its monosaccharide subunits? Number of units, 17. We subtract 1, and the answer would be 16 molecules of water would be needed to split it up into its monosaccharides. So we're now going to talk about polysaccharides and the structural role that they provide. So we're going to talk about cellulose, which we can see down here in the bottom right-hand corner. Looks very different to all the other polysaccharides that we've looked at so far. Now, cellulose is a polysaccharide, but it's formed of beta-glucose. So if we remember from the last video, beta-glucose is the isomer of alpha-glucose. Cellulose forms these long, unbranched, straight chains. Now, this is very important for the type of bonding that it forms. These straight chains allow these hydroxyl groups to be exposed on the top and bottom sides of the chain, and this allows them to form hydrogen bonds. These form between the chains. And when we look at drawing cellulose and how to draw condensation reaction and the glycosidic bonds between beta-glucose, we'll explain a little bit more about these hydrogen bonds. But they essentially form these strong fibres called microfibrils through this hydrogen bonding, and this gives it a very strong structure, and it's used in plant cell walls to keep your plants very, very rigid and turgid. So that really is the only example of a structural polysaccharide that we need to know for this course. Like I said, we'll cover in a minute how we draw it. But another good example to be aware of, just to use as a topic of outside reading for if you end up having to write an essay about this in your exam, is chitin. Now, chitin again is a structural polysaccharide, and it can be found in the exoskeletons of insects and crustaceans, and it's also found in the cell walls of fungi. So in the exoskeleton, it provides this almost armor-like structure for a lot of insects. It's very rigid. And the monosaccharide units that make it up are something called N-acetylglucosamine, which is a horrible word to say, but it's essentially a glucose derivative. And again, the polysaccharide chains that form from these N-acetylglucosamine subunits are long and unbranched, and again, they link together by hydrogen bonding. So that's just another example of a structural polysaccharide that we can talk about. So as we were just saying, cellulose is made up of beta-glucose subunits. Now, because of the position of the hydroxyl group on beta-glucose, this allows cellulose to have some very special properties. Just forgot, I need to add an OH group on the end. This is a very important learning point. You need to make sure that when you're drawing these skeleton structures that you put a hydroxyl group on each side. Don't make the mistake I just did and forget to add one in, because you'll lose marks. So, if we remember beta-glucose from our last video, on the left-hand side, the hydroxyl group is always attached to the bottom stick coming out from the structure, and on the right-hand side, it is always coming out of the top stick. Now, this makes it quite interesting when we draw the glycosidic bond between these two beta-glucose molecules. So, how they bond is essentially in exactly the same way as we looked at when we look at the condensation reaction with alpha-glucose subunits. However, because we have a hydroxyl group coming from this top stick and one coming from this bottom stick, when you draw the bond between them, the best thing to do is put your oxygen molecule in the middle and draw two lines coming down. So it almost looks like you're almost drawing a triangle. You see the stick coming out from the bottom, and then it does a 60-degree turn and essentially comes up to meet this oxygen molecule in the middle. Not the most pleasant thing to draw, but it's very important to remember because you may be asked it in the exam. And as always, we need to label our water molecule that's given off, and we should also be labelling that this is a glycosidic bond. I'm not going to do that because my handwriting is far too messy on a computer. That's an important thing to remember. So to show you again, we have our skeleton structure. Make sure that you label your hydroxyl and don't make the mistake of labelling it on the wrong stick like I just did. There we go. So we draw our bond. We essentially get rid of this OH group on the right hand side of our second molecule. We draw an oxygen in the middle, and we join them up. So it almost forms this quite intricate pattern when you draw it, and then make sure that we label water being given off. Now, something really important to note, that the way that these beta-glucose units bond, that cellulose essentially forms these long branches. The structure is actually not able to bend very easily like we saw when we were looking at starch and glycogen, where they can form these branches and they can form these more spiral structures which are very intricate. Well, with cellulose, what happens is, I'm just going to draw a very basic diagram here to illustrate this. Is that the beta-glucose subunits form these long straight chains. So you can just see our monosaccharide subunits, and we're drawing the bonds between them. So they've now formed these lovely polysaccharide cellulose chains. And what essentially happens is that these chains line up and hydrogen bonds can form between them, allowing them to form this almost solid grid-like structure. Now, the reason these hydrogen bonds can form is because hydrogen is a dipole-dipole attraction between molecules, and so the hydrogen from one beta-glucose subunit can bond to the electronegative atom of oxygen in another beta-glucose subunit. So we get hydrogen bonding between the chains, and these hydrogen bonds are called microfibrils, and they provide really strong structural support for the cells and the cell walls in plants. So to test if starch is present in a solution, we take a sample of the solution that we want to test, and we add iodine dissolved in potassium iodide solution. Bit of a mouthful, but you must say iodine dissolved in potassium iodide to get the marks. As we all know by now, A-level biology is very strict on their marking and their mark scheme, and so you must write that specific term in order to get the marks. Saying iodine, they probably won't accept in their mark scheme, so it must be iodine dissolved in potassium iodide. So we add that, and the sample will turn a browny orange colour when you add it. Now, this is a very quick test, and if starch is present, the solution will then turn this bluey black colour, and that indicates that starch is present within the solution. So to recap what we've covered today, oligosaccharides are sugars made up of 3 to 10 subunits. Polysaccharides form longer chain sugars. Glycogen forms many side chains and is a compact and insoluble molecule, and it's the energy storage molecule in animals. Starch, which is the energy storage molecule found in plants, forms either amylose or amylopectin. So remember, amylose forms these 1 to 4 glycosidic bonds, allowing it to form a coiled structure, and amylopectin has 1 to 6 glycosidic bonds, allowing it to form this branch chain structure. Starch is both a compact and insoluble molecule. Cellulose forms straight chain structures with hydrogen bonding occurring between the chains to form these strong microfibril fibers. And starch in solution is indicated by a blue-black colour with the addition of iodine dissolved in potassium iodide solution. So to fully ensure you've understood this topic, take a few days away from it, refresh it a couple of times over these few days, and then answer the following test questions. Describe the differences between amylose and amylopectin. How does their structure and bonding differ? What type of structure does glycogen form? What monosaccharide makes up glycogen? How does the bonding in cellulose provide structural support? And finally, describe how to perform a starch test. That's all for today. Thank you for listening.